Hello everyone, I'm Stefan Gaugler and I will present you today the fully automated dry blood spot analysis of PEth with the hematocrit correction. For those of you who don't know me from conferences or other activities in this field, I have been working for Camag for 7 years now, but I actually started developing dry blood spot methods in 2012 already, and since then for the last 9 years I actually spent most of the time of my career developing methods and investigating the field of dry blood spot analysis. Also in the field of PEth, we already published several papers, and I guess I can skip the PEth intro in this context. So today I would like to talk about the hematocrit dependent effects we have when doing dry blood spot analysis. So basically, we're looking at the area bias and the recovery bias from dry blood spots. So when we compare two different samples, one with a very low hematocrit and one with a high hematocrit, we see if we take the same volume, the low hematocrit generates a large dry blood spot with a bright color, and the high hematocrit a darker blood spot with a smaller area. As we always take a sub punch with the automated setup, 6 or 4 mm, this can result in an area bias. Another effect is the recovery bias, which says that with a very high hematocrit, for example here, we get a lower recovery than with a low hematocrit. Additionally, we have effects like the volcano effect, which says that the drying at the border zone is faster than in the middle. And we also need to consider the red blood cell to plasma ratio. So we have been working on creating automation for this field. And you can see this basically in the 4 modules displayed on this slide. First, we have a image processing, where the card is analyzed in terms of the label and the spot quality. So we scan the spot location, the size, the area, and the roundness. Then followed by the hematocrit detection. This is a new module which we will launch in April this year. Then we spray the internal standard directly onto the blood spot before going to the extraction cell. The robotics can transport up to 500 cards to the modules, which you can see in the back. First, it will go to the camera unit, where we scan the label and the spot quality. It is important to always extract from the very center of each dry blood spot because of the volcano effect, which I mentioned before. If we treat the calibrators and the samples the same way, this effect is absolutely nullified. We also get a process control of the spot quality and can set this to certain criteria. And here we can see the robotics in action. In the next step, the sample hematocrit is detected directly from the dry blood spot by this optical probe, which is based on a reflectance measurement. First, the card background is scanned, and then we scan the middle of the dry blood spot. And an algorithm which is integrated in our software will then give the hematocrit value, which is based on a calibration line previously set into the instrument. Light at a certain wavelength will hit the DBS, and the reflectance will go to a spectrometer. We detect the sum of the hemoglobin and the oxidation products of hemoglobin. And as we take the sum of these products, we will get a constant signal which is proportional to the according sample hematocrit value. So independent of the DBS age, we will get a proportional signal as we take the sum of these oxidation product. And this was validated and looked quite good against the true hematocrit measured by Sysmex and the predicted hematocrit value. So now we get the hematocrit value, but what are we going to do with it? So basically we are working on a new paper where we establish guidelines to correct the sample results according to the hematocrit value. And here it is important to really standardize the way how the calibrators are prepared and integrated to the system, what reference method we use, and what algorithms will be behind this procedure. The third module is an internal standards sprayer where we can apply internal standards directly onto the dry blood spot. And this allows us to integrate the internal standard prior the extraction, and with this monitor the extraction efficiency and nullify the extraction recovery bias, which we saw previously. This technology was developed in our other field of TLC analysis, and basically we have a sprayer and move the DBS card underneath. In the final step, the card goes in between the extraction cell, where a plunger is sealing a circular area on the dry blood spot, and we pump extraction solvent from the bottom through the blood spot back to the bottom, either in a sample loop, which is then connected to the LC-MS cycle, into a trapping column, or into a vial or 96 well plate. After each extraction, there is a wash station going in between the extraction cell to really wash the whole setup and to prevent carryover. If we now compare this automated extraction with the manual punching of DBS, we see that we have a so-called active extraction. So we always have fresh solvent hitting the DBS, ending up in a very concentrated extract within less than a minute, depending on the extraction volume and flow previously set into the sample list. On the other hand, punching requires much more solvent and more time, and we have a passive extraction, as the extraction efficiency decreases over time. Also, there are several steps involved into the procedure, from punching discs and adding solvent, transferring the supernatant, etc., etc. For the analysis of phosphatidylethanol, we previously published a method which baseline separates the two homologues PEth-16O 18.1 and 18.2. This has been validated in a quite large range with a runtime of 5 minutes per sample, whereas the first one takes 7 minutes as the extraction is not overlapped within the first sample. We were even able to write the Nature protocol with this method, where we described all steps involved into the process. For this application, we use a trapping column setup. Basically, we extract with the extraction pump onto the trapping column, and then we flush the PEth backwards onto our analytical column and then into the LC-MS system. While the analysis is running, we have the rinsing pump here, flushing backwards the extraction cell and preparing the system for the next sample. If we now take a look at the results of the PEth measurement with the according hematocrit value, we can see a clear volume area bias. As the PEth sits in the red blood cells, the more red blood cells, the more hematocrit we have, the higher the signal we get. So this really reflects nicely this area bias which we saw here beforehand. However, there's also an additional effect, which is the plasma to red blood cell ratio, which plays a critical role here. This can be addressed by the two modules I explained previously, by integrating the hematocrit scanner. We can nullify the area bias with an algorithm, and by integrating the internal standard sprayer, we can nullify the recovery bias directly within the analysis process when we take the ratio of the internal standard to the sample signal. So if we now integrate those two modules into our measurement, we can see that the blue line can be corrected to the green line and presenting a much more robust analysis, which is independent of the sample hematocrit value. All goes fully automated, and the hematocrit detection is non-destructive. It can be done prior internal standard application and is prior the extraction. So to summarize, in April this year, with the implementation of the fourth module, the hematocrit scanner, we are now entering an area of quantitative DBS analysis from non-volumetric samples, where we can correct for the hematocrit area bias and the recovery bias by this optical detection module and the sprayer. The protocols for PEth analysis have been validated and even published in Nature protocols, and there is a low error rate through the automation and the integrated documentation system, which really goes into the direction of GLP, good laboratory practice. And with this, I would like to close this presentation and thank you for the attention.